First of all, let me introduce fluorescence polarization. It's a very simple and elegant method to tackle biochemistry and biophysics of different environments. It uses excitation, linearly polarized excitation, and we detect the signals, fluorescent signals, both co and cross polarized with incident light. It happens to be sensitive to local environment, binding, and fluorescence lifetime. So why we decided to try it for fluorescence polarization image? Why did we try to do fluorescence polarization imaging for cancer detection? Is because it's a quantitative method, which is very helpful. So we can just associate a number with presence or absence of the disease. We can do it in real time. It's very simple. It doesn't require tissue destruction, can be used in single cells, and can be implemented at moderate cost. And I would like to emphasize that we are not dealing with endogenous fluorescence polarization. We are dealing with exogenous fluorescence polarization, and we are working with methylene blue, which is FDA approved, positively charged dye. And it has absorption band in the red spectral range and emission band in the infrared spectral range. When I started at UMass Lowell, the first project was looking at fluorescence polarization of methylene blue of breast cancers. And Rakesh Patel was one of my first students, and he has acquired beautiful images showing that fluorescence polarization of cancer is indeed increased. But there were a lot of questions as to whether we really look at fluorescence polarization or it is the signal that is modulated by the optical properties of tissue. So I asked him, can we try and detect increased fluorescence polarization of methylene blue in single cancer cells? And he said, yes, of course, I'll do it. And what he did, basically, he took the images, segmented them, then calculated mean fluorescence polarization. And these were confocal images, so there is no multiple scheduling there. And he saw that fluorescence polarization is indeed increased. Immediately, we put in an NIH proposal, and it came back, not discussed, stating that there must be some mistake here. It's an error. It cannot be. It is not possible. So we published this finding, but not in Cancer Research as the previous publication, but in Journal of Biomedical Optics. And then on board came Xin Fang, who had undergraduate major in biology. So she conducted cell culture studies. I warned her that most likely we'll have no good results and that will be a failed project. But she said, okay, I'll do it. And the goal was basically to confirm that indeed we see high fluorescence polarization in cancer and explain why we see high fluorescence polarization. And several years later, and many few thousand cells later, we confirmed that yes, fluorescence polarization is increased in cancer. You can see yellow cells, those are cancerous, and green purple cells are normal cells. So there is a high contrast here, both in brain and breast cells. She also ran some colocalization studies and found that methylene blue accumulates to a much greater extent into the mitochondria of the cancerous cells as compared to normal cells, and that fluorescence lifetime is much shorter in cancerous as compared to normal cells. So now, with all this demonstrated, we went for the funding again. And what we hear back is, well, that's all really very well. And in cultured cells, there may be such a thing, but you have now to show that it really works in clinical samples. And at that point, now we had to answer the question, can we actually detect methylene blue fluorescence polarization in single cells from clinical samples? For Xin, it was the time to graduate, so she could not help me. And I was really frustrated because I didn't think that there is a single physics student who would be able to conduct such meticulous and such in-depth studies with clinical samples, let alone like handle these samples. But I asked Shin, well, you are training now a student, how is he? And she said, well, he's very detailed. That was his lab book. And I was, oh no. And then I thought a little and I said, oh yes, that's exactly the person we need to do that. And now, several years later, that's all thanks to Peter that we have these results and we recently published them. So now I'll give stage to Peter.
Okay. Yeah, thank you. So let me just bring up the slides that I have.
The lab book very often is a key to something.
Yeah, maintaining the experimental logs is key. Okay, so these are my slides and I think everybody should be able to see them. So yeah, thank you for the opportunity to share a little bit about our recent progress on some of these projects. Great introduction. Thank you for that concerning fluorescence polarization imaging for cancer detection. Thank you, Dr. Soroka, Dr. Franco. So my name is Peter Germain. I'm a PhD candidate in the Advanced Biophotonics Lab. And today I'm going to, as I said, just discuss a little bit of the recent progress using this quantitative optical imaging technique for cancer detection at the cellular level. So the motivation for this project is that epidemiological studies indicate that most adults will develop one or more benign thyroid nodules during their lifetime. And in addition, thyroid cancer poses a significant clinical challenge for the healthcare system. The standard of care for diagnosing thyroid cancer is this procedure called fine needle aspiration cytology, which is subjective in nature because it requires visual assessments of cell morphology in order to identify the disease. So this requires the pathologist to visually identify the cytological hallmarks of cancer in order to identify malignancy, which is very challenging. And as a result, there is relatively low diagnostic accuracy of this procedure, widely varying diagnostic sensitivity and specificity between institutions. As a result, thyroid cytology has what are called indeterminate outcomes in up to 40% of cases. And so what that means is that the cells that you're looking at under the microscope on this slide cannot be reliably distinguished as being cancerous or benign. They're sort of from these gray zone lesions. So that's why we're using fluorescence polarization to interrogate thyroid cancer at the cellular level. And here's how it works. So linearly polarized light is used to excite a sample. In this case, as mentioned, we're exciting the fluorescence emission of methylene blue, this positively charged phenothiazine dye that again is approved by the United States FDA. It's actually a cytological stain that's already used in the clinics and the pathologists are familiar with it. So then you get the copolarized and cross-polarized fluorescence emission images, which, as you can see here, display some morphology of the cells. These are used to generate a fluorescence polarization image, which provides a quantitative measurement for each individual cell. Fluorescence polarization imaging is simple and robust, and that's one of the reasons why our group is using it to explore more accurate quantitative methods for cancer detection at the cellular level. So as mentioned, the current standard of care for detecting thyroid cancer is fine needle aspiration cytology. Thyroid cancer is an interesting disease because unlike other malignancies like say breast or colon cancer, thyroid cancer diagnosis requires fine needle aspiration cytology and cellular level diagnosis as compared to core biopsy and histopathological diagnosis preoperatively. So this particular method is subjective, it's labor intensive, it's time consuming, and it's also relatively expensive, yielding those indeterminate outcomes in up to 40% of cases. Whereas fluorescence polarization imaging permits the single cell analysis, provides quantitative results in real time. And again, we think it holds the potential to reduce the incidence of these indeterminate cytopathological findings. So for this particular study, we conducted multimodal confocal fluorescence emission and polarization imaging of thyroid cells obtained from pathologically diverse clinical specimens. So here you can see some representative images. In the top row, you can see the fluorescence emission images, which these display cytomorphological features of the cells. So for example, you can use them to analyze the nuclear size and shape. What we did is we pseudo-colored the fluorescence emission images from the grayscale images to these images, which have these blue and purple colors to mimic Papanicolaou staining, which is a cytological staining procedures that pathologists are familiar with. And we did this in order to emphasize the morphological features, and we think it will help the clinicians to analyze the morphology in these fluorescence emission images. Whereas in the bottom row, you can see the fluorescence polarization images, which have been pseudo-colored. The colors represent the fluorescence polarization value. So you can see they provide a measurement for each individual cell. And then in addition to that, it's very interesting that they provide distinct quantitative contrast between the cancerous cells in these two left panels, which are papillary thyroid carcinoma and follicular thyroid carcinoma respectively, versus the benign follicular thyroid adenoma cells in the rightmost panel. So for this study, in total, 32 specimens were imaged and analyzed. And what we found was that significantly increased fluorescence polarization of methylene blue was measured in cancerous thyroid cells versus benign or normal. In addition to that, we performed statistical analysis on 12 indeterminate thyroid nodules. And this is, I think, one of the most important findings of the present study is that methylene blue fluorescence polarization provided reliable and accurate differentiation of the indeterminate thyroid nodules. So here's another way to represent the data that we collected. So this chart on the vertical axis is the cellular fluorescence polarization value. On the horizontal axis is the cell size. And here you can see each of the 3,521 cells that we analyzed. And you can see that the cancerous cells, which are represented by these red diamonds, are clustered around higher fluorescence polarization values, whereas the non-cancerous, benign, and normal cells, represented as the blue circles and green squares, respectively, are clustered around lower fluorescence polarization values. And what this graph shows us is that the cellular level methylene blue fluorescence polarization values are not specific to each patient. We can see these general trends of cancerous versus non-cancerous. So just to summarize the findings of this present study, fluorescence polarization cytology may enable more accurate diagnosis of thyroid cancer and reduce the incidence of indeterminate thyroid cytology by an order of magnitude. Clinical implementation of the fluorescence polarization technology can be accomplished without major changes to existing cytopathology workflows. And this approach holds the potential to provide clinicians, including pathologists, a rapid and reliable resource for thyroid cancer detection at the initial fine needle aspiration appointment. So these results were published actually just earlier this month in Cancers. So if anybody is interested, we can definitely forward the link, send the PDF. Of course, happy to answer any questions.
I have a question about this threshold. You put the threshold at 0.245. Is this a threshold just?
That's a great question.
Or did you calculate the threshold?
So this is an empirically determined threshold from the analysis of those 32 specimens containing 3,521 cells. And indeed, what we found empirically is that there were no cells in benign or normal samples that presented the fluorescence polarization greater than 0.245. In the future, and as more data is collected, that threshold may be adapted, and we may use other like statistical analysis methods in order to determine the best threshold going forward.
Unfortunately, what is the specificity and the sensitivity of?
That's a great question. So for these samples that we imaged, out of the 32 samples, so for all of the malignant versus benign or normal sample comparisons, the fluorescence polarization value was greater in the malignant samples as compared to benign or normal. In terms of actual calculating the sensitivity and specificity, I think we would need more samples to do that.
No, I have two comments. One is concerning this threshold value. We didn't aim to find the threshold value.
Sure.
The only thing is that to actually figure out what it will be, we need to very well control the temperature and other conditions, staining conditions, freezing conditions, this kind of thing. But we do believe, based on these results and also cell culture results, plus our confocal images analyzed, we analyze cells and confocal images in tissue, that's approximately the range of fluorescence polarization. I didn't attract attention, but even like what Rakesh calculated for these tissues, the value was approximately that we find here now, but that's at room temperature. And then for sensitivity and specificity, we just didn't miss a single sample yet. But then again, majority of the samples come from either benign or clearly malignant tumors. We need to run the study with this only indeterminate cytological samples.
It'd be valuable to get more indeterminate samples. In this present study, we had 12 out of the 32, but that's one of the next goals, is to conduct a statistically powerful analysis of the indeterminate categories.
So there's another question. So I asked you yesterday or the day before, somehow I'm not a biologist. So the cell size is in between 8 and, I would say, 35 microns.
Yes.
Is this kind of cells having such large variation?
So those cell size values are consistent with cell sizes of thyroid epithelial cells reported in the literature. And there's a wide range depending on whether they're normal or benign versus the malignant cells. I think you can see that the largest cells were indeed in the malignant categories, but these were determined from the confocal images, the cell sizes.
This is clinical specimen. That means that we don't have only thyroid cells here, but also other types of cells. We cannot choose what is pulled out. And that is why when I sent our pathologist, Andy Fisher, the images, he said, like, well, take a look, this is not thyroid cell, and this is not thyroid cell. It's cancerous sample, but it's not thyroid cells. And I asked Peter, can you take a look at fluorescence polarization of these particular cells that are not thyroid? And the bigger one was like something like macrophage or something.
A macrophage, yeah.
and the fluorescence polarization was lower than the threshold. And that is why you see there are no cells which are benign or normal that are above 0.245. But at the same time, in cancerous samples, we have some cells which have fluorescence polarization lower. And we do believe that for the most part, these are not thyroid cells or not cancerous cells.
So
Because we
that,
don't have a choice.
But this can be stained so that you can identify.
Yes, you can
Yes,
show,
and
she
yeah.
can show you fluorescence
so one
images.
thing, so the samples all were lysed, so to eliminate erythrocytes from the samples. And then indeed, that's correct. The fluorescence emission images provide this capability to analyze morphology so that when we were performing the analysis, you can usually look for cells that have, like, say, a clearly defined nucleus, and you can select the thyroid epithelial cells for the analysis.
Because our pathologists were as suspicious, even more suspicious than you are. When they looked at our data, they were just like, we need to make sure that.
I'm not directly in this field, but there are coming up some more questions.
Yeah, please ask.
You expect a clear separation. Can you go back to the previous slide, please?
Of the
Yes.
scatter plot.
So you remarked that some of the red dots that not cancer cells may not actually be cancer cells. So if you have the ability to separate these manually and rerun the experiment, would you expect that there will be a clear separation between cancer cells and benign or normal cells at 0.245?
It's our dream. We hope it will come true.
Good dream.
Yeah, it's an interesting question because previous studies from the Advanced Biophotonics Lab indicate that the cellular level fluorescence polarization measurements are sensitive to mitochondrial dysfunctions in cancer cells as they progress in their development to malignant status. And so I'm not sure if you would expect to see clear delineation versus some gradient as the cells.
Become more malignant.
Yeah.
No, we didn't look at, the goal is basically to take maybe 30, 40 cells as is done now and see if some of the cells have fluorescence polarization that is higher and then confirm by tissue histology that indeed these are malignant. So that this could be used in clinic for rapid assessment of FMAs. So that's our immediate goal.
Mm-hmm. Yeah, and as we go forward, now that this feasibility study has been conducted, we're also working on enhanced sample handling, cell smearing protocols to sort of further the translational development of the technology so that it's something the pathologist would actually want to use.
So what is the actual concentration of methylene blue which you apply for this? So you make a protocol for this, and this is optimized for thyroid cells?
Yeah, that's a great question. So the staining concentration was 0.05 milligram per milliliter methylene blue. The actual concentration in the cell environment would vary.
We don't know, we never measured. We just know that that was from cell culture studies that Shin performed. We know that from 0.01 to 0.05,
0.07.
this concentration give us good results. We haven't tested anything outside. And then Peter took Shin's protocol and applied directly because we didn't have funding or time to just fiddle with optimizing concentrations. Now we're in a better position because we know it works. Now optimization makes sense.
So the FDA approval for MB is in molecule, so in the concentration, to apply concentrations much more higher, so 10 times, 100 times higher.
I think I'm trying.
100 times.
Yeah, I'm trying to remember. It's like 1%. It's like
10
10 milligrams per
milliliter.
milliliter is approved to guide,
And
for
they
example,
counted,
sentinel
like,
node
it's
biopsy.
like
And how long does it take when you take the aspirate the cells. So what is the protocol at the very end for the pathologist? So they aspirate the cells, put it on the plate, and then you put the specific concentration of MB on it, and then you wash it a little bit or not.
You can
And,
wash the
then you make
sample
the
to remove the
washing
excess dye.
to rinse all the maybe the methylene blue in the suspension the way that you only can characterize the cells, right?
Yeah, we rinse 3 times.
Okay,
To
That is in one
remove
of
background, yeah.
okay. To reduce it.
But that's what we're currently working on,
And
trying to
how
have the
long
most.
does it take this procedure? So the incubation of the MB.
So currently the staining time is 20 minutes. The plating time, we're hoping to reduce by using a new protocol. So instead of say like plating the cells, we're developing like a smearing technique using the methods that are currently used for like similar to rapid on-site evaluation of cytology samples. For this particular study, what we found to be the most time-consuming step in the analysis was the manual segmentation of the cells in order to perform the quantitative analysis. But another graduate student in the lab is also has developed a MATLAB program that permits auto segmentation and auto calculation of the fluorescence polarization values. And we have really some really interesting preliminary results that show that the results from the auto segmentation and automated generation of the F-pole values correspond to the manually processed values to within, I think, like 0.5% or something.
I have a question,
Sure.
and it's about the mechanism. So this is methylene blue, right? That's
The fluorescence of
right,
methylene
that
blue.
is uptake by the cells. So what is the mechanism?
That's a great question.
So you can go to my presentation, it's on and explain the results.
Oh,
with,
sure.
With lifetime and localization. I just didn't talk about it, but I think Peter can talk about it.
So maybe it's like, is the screen sharing? I think it's sharing. It says.
It says stop share.
So yeah, previous investigations that were performed in the lab and the work was conducted by Xin Fang and investigated this idea in the cultured breast cells and the cultured brain cells about like what is the mechanism of the increased fluorescence polarization value in cancerous cells. And so here's some of the data. So I'm just trying to make sure I start in the right spot. So this particular slide demonstrates that methylene blue preferentially localizes to the mitochondria of cancer cells. So there's greater uptake of the methylene blue dye in the mitochondria of cancer cells as compared to benign or normal cells in this case.
The 2 upper graphs, they show breast cells at
Okay, yeah.
different concentrations, right? The results of localization for the breast cells. And the bottom graph shows same analysis for brain cancers.
Okay, so I think so what you're hinting is that it's a metabolic mechanism, like because going to the mitochondria, right? And then so then my question is, if you have the same cell line but at different metabolic states, will you have different optics?
You know, that is we have more simplistic explanation because methylene blue is positively charged and mitochondria are negatively charged. That's why we have preferential accumulation in mitochondria. But the cancers we're looking at, they also have increased mitochondrial membrane potential, like thyroid, cancers, brain, breast. That's why there is even higher accumulation of methylene in these cells in this cancer.
And what about the transport through the cell membrane?
That's why we need low concentrations.
But is this an active transport or is this diffusion? This cannot be explained by the charge. So because this is too far away.
Judging by the time it takes for staining, it's probably diffusion.
It's a question. So
But again, membranes are also negatively charged. So some dye would stick to the membrane as well, but I would say it's diffusion. But yeah. And then another explanation is the next slide with the fluorescence lifetime.
So yeah, this particular slide showed that for cultured breast cells, the fluorescence lifetime in cancer cells is shorter than in normal cells. And because the fluorescence lifetime is shorter, there's less amount of time for rotational diffusion of the fluorophore during the lifetime of the excited state, which preserves the polarization of the fluorescence emissions relative to that of the normal cells.
I think all the measurements are fine in direction that or thinking that the MB is within the mitochondria and so on. But this is not a explanation for how does the methylene blue transfer through the cell membrane and then at the very end accumulates to the mitochondria. So my question is about the transport through the cell membrane, whether this is an active one or a passive one or whatever.
That's a good question. I'd have to look more into that.
But I think it should be mentioned somewhere in the literature.
Yeah, we didn't look into it, and it is one thing that is clear that it's not that methylene blue accumulates only in mitochondria. It's not specific to mitochondria. It definitely stains nuclei, all nuclei. Normal cancers, that's why it's used in pathology as a nuclear stain. It also goes into the lysosomes. And in our publication, which is open access, and I'll send the link, we explored all three sites, nucleus, mitochondria, and lysosomes. And with nuclei, we saw that methylene blue goes to all the nuclei, irrespective of cancerous or normal. With lysosomes, we saw a trend that it accumulates more to cancerous lysosomes in breast, but we could not confirm it in brain. The only organelle that always has a lot of methylene blue was mitochondria. That's why we kind of stick to this explanation. But yeah,
This is also a biological question, and it seems to more or less physicists.
Yeah.
oh yeah, that's a good approach. Just let somebody else.
Yeah, but I guess my question, if you don't think of this as a cancer tool, but more like a metabolic assay, would you have differences in cell outcomes based on metabolic activity?
And so one of the things too, and we're working on and still analyzing, but so we're also doing like imaging and analysis of the breast FNAs, and we have performed some preliminary analysis of the fluorescence polarization versus the grade of the tumor, and have found like some interesting results. So there may be a correlation of like higher fluorescence polarization in grade 2 versus grade 1, and so we are looking at that.
And also, yeah, it's good that you recall that, because
Yeah.
there was a big part of the study with breast to see if we can also target aggressiveness, because there is this huge gray zone in breast cancers where you don't know whether it will kill or person will survive and die of other courses like 50 years later. And we looked at the aggressiveness, Peter did look
The
at
tumor
it,
grade and aggressiveness, yeah. And it's like, from my understanding, as I was just been like looking into this too, is that sometimes for the higher grade tumors, that you get the Warburg effect, where you shift from oxidative phosphorylation, which occurs in the mitochondria, to glycolysis, which occurs in the cytoplasm. So that might also provide some like interesting shifts in the fluorescence polarization in these high grade tumors. So yeah, looking into that.